Our brain is made of billions of special cells called neurons that work together to allow our body to do everything we need to do to survive. Instructions and information are passed from neuron to neuron through a series of rapidly moving chemical and electrical signals known as action potentials. Before we look into the mechanisms and structures behind an action potential, let's have a closer look at the neuron. First, we have the cell body in red. This is the largest structure of the neuron. Coming off of the cell body are the dendrites, which receive incoming signals from other cells. Inside the cell body is the nucleus. As with every other nucleated cell, this acts as the control center for the neuron. In blue is the axon, which is the long, thin part of the neuron extending from the cell body. The axon carries electrochemical signals from one end of the neuron to the other to be passed to the next neuron. Shown in yellow are the myelin sheaths. These layered fatty sections increase the speed with which the signal is propagated through the axon by providing insulation. The myelin sheaths are produced by Schwann cells, shown in orange. The spaces between these sections of myelin sheath are known as nodes of Ranvier. These allow ions to access and to pass through the membrane of the axon. Lastly, the axon terminal, shown in green, branches off from the axon and releases vesicles containing chemical signals known as neurotransmitters into the synapse to be taken up by the dendrites of the next cell. Now that we know a little more about the neuron, let's talk about another important concept: gradients. Shown are two areas separated by a permeable membrane. On one side, there are a lot of particles, and on the other, there are only a few. This is a concentration gradient. Because nature has a preference toward equilibrium, this gradient will cause particles to move from the area of high concentration to the area of low concentration. A real cell membrane is only selectively permeable, though. Charged particles cannot pass through it unaided, so special membrane proteins are required to allow them through the membrane. Channels are proteins that allow ions to flow along the gradient across the membrane. Pumps use energy to move ions across the membrane going against the gradient. An action potential is an electrochemical signal that moves down an axon. Membrane potential refers to the difference in charge of the internal and external environment of the cell. The resting membrane potential of a neuron is negative 70 millivolts, meaning the external environment is more positively charged than the inside of the cell at the axon. As a response to sufficient stimulus, the membrane potential will become positive in one section and trigger the membrane potential of the next section of the axon to become positive as well. This continues as a chain reaction until the electrical impulse reaches the axon terminal, triggering a neurotransmitter to be released into the synapse, which is the space between two neurons. When you take a closer look at the axon, you can see how concentration gradients and membrane transport proteins allow action potentials to take place. The pink squares represent positively charged sodium ions, and the purple dots represent positively charged potassium ions. As you can see, there are a lot more ions outside of the cell than inside of the cell. Some stimulus causes the internal cell environment to become more positive. This change in membrane potential, measured in voltage, causes voltage-gated sodium channels to open. Because of the charge gradient, sodium ions will flow into the cell. This causes the concentration of positive ions inside the cell and outside of the cell to get closer, which is called depolarization. Once the membrane potential gets high enough, the sodium channels close and voltage-gated potassium channels open, allowing potassium ions to flow out of the cell. Because there is more potassium inside of the cell than outside, the concentration gradient causes potassium ions to spontaneously flow out of the cell. Potassium ions also flow out of potassium leak channels, which are always open. This process is called repolarization. As the action potential moves on to the next section of the axon, The potassium channel stays open, and the membrane potential dips below the resting potential to prevent the action potential from moving anywhere but forward. This process is called hyperpolarization. The time during which the cell is hyperpolarized is called the refractory period because it allows that part of the cell to return to its normal state. Resting potential and concentrations are reestablished by sodium-potassium pumps, which use energy to move sodium ions back out of the cell and potassium ions back into the cell. When an action potential reaches an axon terminal, it triggers the opening of voltage-gated calcium channels that allow positively charged calcium ions to enter the cell. These ions interact with vesicles containing neurotransmitter and cause them to fuse to the internal membrane and release the neurotransmitter into the synapse. The synaptic structure shown is cholinergic, which refers to a synapse through which acetylcholine travels. When acetylcholine reaches the postsynaptic cell, it binds to the acetylcholine receptor, which is a ligand-gated ion channel. That simply means that it only opens when a ligand, acetylcholine in this case, binds to it. This channel lets sodium ions into the cell to start a new action potential in the postsynaptic cell. Any remaining acetylcholine in the synapse is broken down by acetylcholinesterase, and choline is reabsorbed into the presynaptic cell through the choline transporter.